Dear students, please note down. The PDF file of all these notes have been uploaded in the description of video lecture. Please download your notes. Dear students, today we will discuss Cori and Cori cycle. This Cori cycle, which is an important topic of muscle physiology. This is final exam topic for BSc students. Dear students, this Cori and Cori cycle, it was proposed by C.F. Cori and Theresa Cori, both husband and wife. They got Nobel Prize along with Housey in 1947. This Cori cycle was given by C.F. Cori, husband, Theresa Cori, wife, and they got Nobel Prize along with Housey in 1947. Dear students, this Cori cycle, it occurs in muscle and liver cells. As we know, the food gets stored in the form of glycogen in our muscles and liver. That glycogen in anaerobic respiration, during strenuous exercises, during vigorous exercises, that glycogen undergoes glycolysis, anaerobic glycolysis, EMP pathway. Dear students, Embden-Meyerhoff-Parnas pathway. In that process, one glucose molecule, that is a 6-carbon compound, and this 6-carbon compound is broken into 2 molecules of pyruvic acid, 3 carbon each, and 2 molecules of reduced coenzyme NADH2, as well as 2 molecules of ATP are generated. This pyruvic acid, it undergoes hydrogenation. As a result, this 3-carbon compound, pyruvic acid, it is converted into 2 molecules of lactic acid in presence of reduced coenzyme NADH2, and this reaction is catalyzed by LDH, lactic acid dehydrogenase enzyme. It means in anaerobic respiration, at the time of vigorous exercises in our muscles, the stored food, which is glycogen, and that glycogen is converted into lactic acid. Accumulation of lactic acid in muscles results muscle fatigue. Dear students, it occurs in glycolysis, EMP pathway. In that process, this glucose, which is 6-carbon compound, and this 6-carbon compound is broken into 2 molecules of pyruvic acid, and this pyruvic acid undergoes hydrogenation to produce lactic acid. In presence of this lactic acid dehydrogenase enzyme and reduced coenzyme NADH2. Now, this lactic acid, which is present in our muscles, it some part re-enters in the relaxed muscle fibers. So, muscle glycogen is resynthesized, while the maximum part of muscle lactic acid, that is carried via blood into liver. Dear students, muscle glycogen during strenuous exercises, anaerobically, it is converted into muscle lactic acid in glycolysis in EMP pathway. And this muscle lactic acid, it is carried via blood into liver. In liver, about 1/5 part of this lactic acid that gets oxidized into CO2 plus water plus energy. And this energy which is released in this oxidation process, that energy is utilized in the resynthesis of glycogen. It means the maximum part of lactic acid, 4/5 part, about 80% of lactic acid that gets synthesized in liver glycogen. This is Cori and Cori cycle. Dear students, please note down. As we know, the food gets stored in our body in glycogen form, and that glycogen gets stored in our muscles and liver. In muscles, the stored food which is glycogen, and this glycogen undergoes anaerobic breakdown during strenuous exercises, vigorous exercises. That muscle glycogen in that glycolysis process, EMP pathway, it converts into lactic acid, and that lactic acid, it is carried by the blood into liver. In liver, about 1/5 part of lactic acid gets oxidized into CO2 and water, while the maximum part of this lactic acid, about 80%, that is 4/5 part of lactic acid, and that lactic acid gets synthesized into liver glycogen. This liver glycogen undergoes glycosenolysis, means breakdown of glycogen into blood glucose, and that blood glucose carries this glucose into muscles. Here in muscles, resynthesis of glycogen takes place. It means in muscles, glycogenesis means synthesis of glycogen. As a result, this blood glucose gets stored in the form of glycogen in our muscles. This is very easy topic for BSc students, Cori and Cori cycle that takes place in our muscles and liver. Dear students, this entire process was given by C.F. Cori, husband, and Theresa Cori, so it is named Cori and Cori cycle. In next lecture, we will discuss electrical and biochemical events of muscle contraction. Thank you.